Alright, YouTube's being mad annoying. I can't stream, so I'm gonna just have to record these videos and have it all saved on my PC. Man, what are you gonna do? That's fine, as long as I get studying done. Okay, I'm gonna start the timer. Oh, I don't even have the slides ready, so these are my slides. I have just 9, 6, 10. I have 10 slides. I just have to go through all of them. Let me go from dynamics. So I'm gonna go from top to bottom because these are like fresh in my head, and then yep, let's do that. It's like load. That's what I'm gonna do. I'm gonna have this on a separate doc, and I could switch like that. And I'm gonna just make notes. Put it in Helvetica. I like Helvetica both. I think Helvetica both fire. So, dynamic cell. And I'm only going to write down what I might not remember on the exam and what he probably would ask. Honestly. All right. Ready, set, go. Oh, we already started the timer. Let's go. Okay, let's turn that off. Turn that off. Okay, let's go. Some okay, then coronavirus, spike protein. Obviously, we can grow, reproduce. It's life. 200 micrometer. How big is the zygote? All right, we can copy this down. I would say zygote. I'm gonna just copy down numbers, bro, like little number shit. Okay, I'll clean it up later. I'm just trying to get everything we need. Living system. All right, what I remember. Organ system, then tissues, cells, atoms, molecules. Did I get that right? Hey, something like that. To understand multicellular single cell, basic unit of life. Okay, all cells have the same thing. Biochemistry, all this crap. They move and stuff, or they develop, they operate, they communicate, they walk around, and what else? Control activity. Different. Okay, they have different sizes. You have RBCs look like that. Plant cells look like that. Purkinje neuron. I mean, if he's not gonna throw a fucking picture of that stuff. Okay, this is important. I think he might ask a question on this, like how, the size and shape. So I'm just put size and shape. Extreme bacterial cells. Yeah. And yeah, he might ask this. I think this is like it's like a simple question he might ask. How big is how bacteria cell? Okay, divided in two groups. I think it's eukaryotes and prokaryotes. Remember on organelles? Okay, yep. Complain. Okay, what does eukaryotes? I think eukaryotes is important. I never remember what a eukaryote is. Let me try to get this to Helvetica Nui. I don't like the bold. I don't need the bold right now. Eukaryote. I need to move quick, bro. This is a waste of time, honestly, going through this chapter. You're the goat. Protist. And then, I never remember this, honestly. Eubacteria. Oh, I think we should use bullet points. He will not ask that chart. Stupid chart. Yeah, eukaryotes, eukarya, eubacteria. We don't even go over that in class. All right. Nucleoid. Yeah, I know they have a nucleoid. They don't have a nucleus, so they have a nuclear region. All right. Yep. Compartments. Examples of compartments. Examples of compartments is a pretty good question. I'll just make my sister like go over this with me. Make sure I remember it. Maybe just delete it if I know it. All right, compartmentalization. Yeah, so make sure you get like concentration and stuff. Was it twofold change in diameter? Oh yeah, the bigger you are, the more freaking enzymes you need. That's a pretty normal thing. Okay. What is it? Two examples: compartmentalization. Chloroplast is for like photosynthesis, I think, and then mitochondria is for ATP, phosphorylation, all that stuff. Oxidative. Okay, yep. I remember that. Most tubules, microtubules. Okay, what I remember, right? I know cytoskeleton is made up of microtubules. It's made up of microfilaments. It's made up of intermediate filaments. Microtubules, bro. I took notes on this too, bro. I remember microtubules is like alpha and beta tubulin, and it's like travel on intermediate filament. It's like very resistant, and then intermediate. I know intermediate is like very resistant, and then microfilament and actin and myosin. Yeah, bro, we gotta remember this. Contraction will. Yeah, this is. I just don't remember this, bro. This is just stupid. Muscle fibers. It's just like a stupid question you might ask, bro. Like, oh, what is a microfilament? Oh, what is it do? And I just won't remember it, bro. Okay. Yep, this is good. And what is the order? I remember the order. I think intermediate is the middle, microtubules is like the top, and then microfilaments is the bottom. Cytoskeletal, motility, contraction, motility. Okay. Eukaryotic cells, endocytosis, invagination, and then vesicles form, and then they get the hell out of there. Right? That's it. That's pretty simple. They don't have it. Okay, remember you can remember prokaryotes don't have endocytosis. Endo or exocytosis. How much time do we got on the clock? We got 39 minutes. Hopefully I can get through 3 chapters in that time. Okay, imagination. Okay. Yeah, this is something I can remember. Prokaryotes, circular DNA. I know that's circular DNA folded into a... Okay, this is something I remember. E. coli is... I'm going to just put like a whole prokaryote thing right here. Don't have... Coli, 1300. I know it's circular DNA. She likes on you to go. Alright, eukaryote. See, it's just like little stuff like this. Oh, does it fold more or does it fold less? And then I didn't remember. Okay. Nope, I don't even know that. I mean, just look at it, right? Mitosis, meiosis, prokaryotes. How do replication of DNA followed by cell fission? Okay. So you replicate and then okay. Replicate and then cell fission. Replicate DNA. Okay. Yeah, I never remember the cell cycle, bro. I think I should remember it somehow. It's not that hard. It's really not that hard. I'm tripping. Mitosis, meiosis, and distribute cell division. Okay. Cell cycle. Cell cycle. If I remember correctly, this is how it is. It goes like G1. G1. I would just actually have like and then S, and then G2, and then M. G1. I think everything is like dividing. Just write it down. Remember it. RNA lipid. He won't even ask us, bro. It's such a stupid question. But I think I should remember regardless. I think this is just something important to remember. DNA replication. Okay, DNA replication. DNA replication. More synthesis, mixture, cell mixtures, nothing got fucked up. As like drink. For errors and stuff. Basically, it's like monitoring. Make sure nothing gets divided. Okay. Yeah? No. for errors. Fixes. Okay, this I should remember. Fixes damage. Alright. Last thing, I think is cell division, right? Cell division. In 2-2. I think, is that my total miles? It's, I can remember that. G0 is, okay, let's move on. Yeah, no, they don't have introns, so they don't get spliced. There's no nutrients, so they just like, it's like co-transcriptional for them. What else, bro? What else? That's it, right? Difference in the sizes. Yeah, I know they have like smaller ribosomes too, right? This is prokaryote. It happens automatically. Larger. Okay, what is it? Activation, and then transcription happens. All right. What's the order, right? Addition of the cell methyl guanosine cap. Okay, after the cap is there. After the cap is there, what happens? Do we splice out? I can never remember the order, bro. I think this is such a stupid detail too. The cap is down, and then we remove introns, and then we cleave the site, and then we add adenines to it. Nah, that was right. I actually got that pretty right. In short transcripts, since they're short, you're gonna remove the introns after polyadenylation. But if it's long, since we don't got time to wait, we'll probably do it, coach. We'll do it while polydation happens. Yep. All right, this is good. Not reading all that. Okay. Eukaryotic cell. What are the main things about it? It has a plasma membrane. It has a nucleus. It has membrane-bound organelles. That's chloroplast, Golgi. Better get that. Cytoplasm. Plants have a cell wall. I mean, I knew that. Phospholipids and proteins. We could write that down. Glycolipids. Dude, I'm taking too long, bro. Glycoproteins. Yeah, I know what they do, bro. I know that they are receptors, bro. Nucleus. All right, let me just everything. I'm just gonna dump everything I know about the nucleus. Nucleus is made of a double membrane organelle. The interior is made of nucleoplasm. There's a nucleolus. Is where RNA is where like pre-RNA, rRNA, like RNA, like ribosomes and stuff are made. It's the control center. It runs everything. Has DNA. The chromosomes are located in the nucleoplasm. Control shit. The pores, right? They have pores that transfer stuff. And nucleol. Yeah, that's it. Everything, right? Okay, we're good. RNA enzymes. Yeah, everything gets inside. Two-dimensional. I know protein lamina, which is that's a good question. What is it? Intermediate filament. Lamina equals intermediate filament. I would not remember that. Actually, it's pretty strong, right? The government study was okay. Alright, obviously the pancreatic acinar cells, they secrete enzymes. These are proteins. Obviously, you're gonna have to leave the cell, so you're gonna have to go to the secretory pathway. Alright, mitochondria. Of the size of the bacterial cell. Okay, I didn't know that. I'm gonna just write that down. Mitochondria equals size of bacteria cell. It's like a little true or false question. Contain their own ribosomes and DNA. I mean, I knew that much. Write that down, right? Synthesize some of the proteins. Most activated hormone in the mitochondria. Okay, that's pretty obvious. Anything more about it? I need to know. Cristae, I know that the enfoldings harbor most of intermediate as well as okay, matrix. TCA cycle, fat oxidation. Okay, okay. This is I never remember this, bro. The TCA cycle occur in the matrix. Christe, matrix, general matrix. Okay. Mitochondria. You're gonna have more of it when you need more energy, so you're gonna have a lot of it in the sperm head, right? That's what I said. Oh, is it not in the sperm head? A sperm cell around the central shaft. Oh. Why am I writing like that? Oh, wait. Come on, baby. Lots of mitochondria in central shaft of sperm. Axonium. Axonium. Oh, sperm. A chloroplast. Size of it, I never remember, bro. I don't remember the size of these things, bro. Sorry. Chloroplast. How much time do we have? 20 minutes. Okay, hopefully I can finish this by in 9 minutes. Large organelle. Smaller only than the nucleus, so very large. Second. Found in leaf cells and other photosynthesis. Okay, that's obvious. Surrounded by inner. I know it has two. I know it has 3 members. Thylakoid. Okay, so what I remember is that, yeah, the stroma, yeah. I know that's what happens. Okay, carbon dioxide, water. I never remember what photosynthesis is. Come on, give me the reaction. Someone, thank you. I never remember what this is, bro. Carbon plus water gets you glucose and oxygen. I don't even know what light dependent means. Okay, move on. Okay, I know cistern A. What's the order? Cis and trans? Interconnected. That was lipid steroids and activation. So you're gonna have a lot of. Okay. Camilo Golgi. The Golgi was named after Camilo Golgi. Camilo Golgi. Nah, Camilo Golgi. Such random information, bro. I don't not. How are we supposed to remember that it makes complex polysaccharides, bro? Yeah, budding. Lysosome. Very small. Very small. think I know that though. Oh, I should have just said it out loud, bro, so I could just remember. I don't know why I didn't do that. Lysosome is 0.5 to 1 micrometers. It has one membrane. It is going to have hydrolytic enzymes. The pH is going to be around 4.5. And so when stuff like fags and like stuff that enter the cell, it's gonna like bud with them, and it's gonna just destroy everything with its hydrolytic enzymes. Is there anything I'm missing? Hydrolases. Oh, it's gonna break down all macromolecules. Hydrolases. I remember asked my teacher a question. Hey, professor, what's the lysosome do? He was like, you're an idiot. You know, don't ask me that question. You should have read. And then I never forgot since that day. The enzymes, the lysosome. Obviously, made an ER inactivated when entering. Golgi activated from Golgi, like get combined to make lysosome. Is that it? Transported, activated Golgi, and closing lysosome. Yep, that's already it. I think primary lysosome is just that lysosome that comes from the Golgi, and then the secondary lysosome is when that lysosome, like, it's a circle. Like, I have a circle right here, and then you have like shit, and then it just like eats it. Yeah, that's pretty true. Yeah, right. You see the budding. Right here, right here. Lysosome. Food vacuole, second vacuole. I didn't know they were called microbodies. Microbody. You know my body. You know my body. Might remember it like that. You know my body looks like. I know it looks like lysosome. You don't need to tell me that. Convert tox. Oh, I know what they do. I know peroxisomes have enzymes that are called oxidative. They have oxidative enzymes, so they're gonna do oxidative reactions, and that's gonna create like hydrogen peroxide, and then you're gonna use that catalase to obviously break down all those ROSs into water and oxygen. Did I not just say all that? You could say they're found in the liver. I'm gonna go like from the beginning to the end and just like so I get best of both worlds. So I'm not just focusing on the beginning. You know, I feel like that's a good strategy. Yo, I'm really trying to get a freaking 100 on this exam, so I'm doing this whole rigmarole. I know I break some very acids. I never remember this. Glyoxysome. I was like, bro, he's not gonna ask this. Leaf paroxysm. I was like, bro. Now I'm getting bored, bro, because this. Alright. Vacuoles. Membrane-bound organelles used for central vacuums. Like vacuoles, bro? Like... Why not just study like the exam for the... Why not study like this for the first time? I would just saved a lot of time. Mature plant cells have a large mineral. Okay, I can just remember that. Plant cells are main function of central vacuole. Central vacuole maintain turgor pressure. Need to move on, bro. This is how long? Oh my god, we're not even halfway through. Contractile vacuums. I know that the measured in the sedimentation coefficient Svegberg. I don't. To this day, I don't know the difference between the cytoplasm and cytosol. I think cytoplasm is everything, and the cytosol is just that liquid. Protein synthesis, fat synthesis. Oh, I know protein fat synthesis. Okay. Golly, bro, this is a lot. I think the last slides are really stupid, though. I don't need to go through it. How much time do we got? 17 minutes. This is taking too long. Microphones. There are various amount of proteins. We don't know all the proteins that make it up. Come on, you already know this is the best place ever. I know it's polymers. Alpha, I know it's alpha. The four major macromolecules are fats, sugars, amino acids, and carbs. And the macromolecules for them are polymers. Oh yeah, lipids, proteins, polysaccharides. Oh man, I didn't know that. I thought. I didn't know that. forgot about that Shine Dalgarno sequence. That's a sequence that the freaking Procaria Rivalus binds to. That's crazy. Half a billion years ago, we have the same. Like insects and stuff. That's Pack six. Isn't that Pack six? Zygote is 200 micrometers. Body, building cells, about cells. What is it, like totipotent and then pluripotent and then multipotent? Pluripotent, you have ectoderm, mesoderm, endoderm, and that's gonna like specialize into like other things, and then those have specific functions. Implant a nucleus into an enucleated. I think it's the last chapter. I think I know everything. done, baby. Let's go. Chapter 1 is done. Let's go. Let's do a quick review. Sight.